Hello, dear students. Today I'll talk about plant biotechnology. So, plant biotechnology is nothing but it is a process in which people are going to introduce the transgene into a plant cell and develop a complete plant cell, and even they'll go for culturing the plant cells in order to get the commercially important protein molecules, or even to get some small molecules, secondary metabolite productions. Even people are doing it in order to get uh, different type of plant variety for improvement of crop, for pest resistance, tolerance. So many aspects people are actually working on in case of plants in order to improve the quality and quantity of food as well as the plant varieties. So we'll start with the plant biotechnology by going through the process of tissue culture, which most of you have good idea about. So tissue culture, because the plant cells are the one kind of cells which have the totipotency. That means a single plant cell can give rise to a complete individual plant. And the growth and development of the plants means here the plant seeds, organs, explants, tissues, cells, and protoplasts in nutrient media in a sterile condition comes under as plant tissue culture. We come across with here the word called explant, which is the original tissue sample which is taken out from a particular plant. So how it is done? People are going for the process is like they are going to take the explant, they are going to clean it, wash the explant, then they are going to disinfect it, and followed by that it is going to be transferred into a sterile medium. So for that they need to have mm, pre-treatment with gibberellic acid, cytokines are all going to be done. Then we come across with uh, culture medium, addition of oxygen, cytokines. The culture mediums, like there is different type of culture medium is there. MS media is there, DKW media is there, BTM media is there. There are many other culture media is there, which contains micro and macronutrients, hormones, amino acids, vitamins, which are all required for the growth of the plant cells. So then they are going to give specific ratio of auxins and cytokines in order to get the root and shoot inductions. Okay, so increased cytokine concentrations, low amount or absence of auxins are going to be given for 4 to 8 week subculture intervals, so that we can get the root. Then followed by that, after getting the rooting, people will try to means they are going to go for this in vitro rooting. Okay, so decrease the cytokines, increase the auxins. First, increasing cytokines and decreasing the auxin, they'll get shoot, and then increasing the auxins and decreasing the cytokines, they get the rooting. So once the root is also going to get developed, so then they'll go for this one. So generally, when the cells of plants are going to be grown, so that goes into a mass of cell which is called as callus, and in the callus there will be number of nodes. Nodes means they are nothing but group of cells which are going to be present within the mass of these cells of callus. So complete callus can be used to develop one particular single organism plant cell, a single plant, or even these particular callus can be get separated into these different nodes, and each of the nodes can be further developed into a plant body by inducing the shoot and root. So once the shoot and root is going to get induced and they are going to be grown, then after the rooting, so they go for transferring that particular plant from the synthetic culture media, semi-synthetic culture media into into the natural environment. So for that they are going to go for transfer into the natural environment happens in such a way that it will happen first in the in vitro condition. So there will be plants are going to have little of time is going to be required for them to adjust the change in environment, as because of which we can find low photosynthetic activity, water loss control, poor water loss control, abnormal stomata functioning, all these kind of things. Then but later, once the plants are going to get completely acclimatized into the natural environment, so then the plant is going to be grown completely into the natural environment. So this particular method, we can call it as the micropropagation method. That's very very useful in case of plants which are actually having a low natural rate of cloning, means multiplication, and in case of plants which are having high demand and valuable plants like orchids, people are doing. In case of banana, they are doing. Difficult to root the plants, and as well as in plants which are endangered species, and even the plants that cannot be clonally propagated in any other ways, they are being propagated through the micropropagation method. There are a number of advantages, disadvantages with the tissue culture. So this is how the whole process of tissue culture or micropropagation is going to be done. So whenever we are going for this tissue culture, the people can also go for, instead of developing complete individual plant, the people can also go for while doing the tissue culture, they can go for introduction of new characteristics into the plant varieties, or they can make this particular plants to undergo some kind of alterations, hybridizations, etc. So in order to introduce a new gene into the plant, that can be done artificially by the genetic engineering process, or people also do it by the process of somatic hybridization. So for doing this particular somatic hybridization or genetic engineering, what people actually required is a plant cell without cell wall. So because the cell wall, as we know, it is going to be an hard outer covering of the plant cell, which is going to be highly difficult for the transfer of gene into the cell. Means in case if we are going for a genetic engineering process, if we want to go for an hybridization like somatic hybridization, so wherein fusion of the cell is not going to happen if the plant cell wall is going to be there. So therefore, whenever people go for this plant tissue culturing process, when they are going to develop this explained material into a mass of cell, so that particular mass of cell is going to be treated in such a way that the cells are the plant cell wall which is going to be there around the plant cell is going to be subjected for degradation, so that only the plant cell with only membrane is going to remain, which we call it as the protoplast. So once people actually get the protoplast, then they are going to, these protoplasts are not going to maintain for long time as such. Generally people are going to do means the protoplast, once it is generated, it is maintained in a sugar solution, in a sucrose solution, or people are going to use some other kind of osmolyte molecules which can able to stabilize the plant cells. But the plant cells, the plant protoplast will not remain like protoplast for long time. So as soon as it requires means it will get the nutritional substances immediately. So within few hours itself, plants is going to develop the cell wall again, because if not, it will get die off. So once it develops the cell wall, these particular cells are going to get converted into callus culture, and then further it's going to be induced with root and shoot to give the plantlet. So protoplast culture is, as I told you now, like protoplast culture is very very important for genetic manipulations. So whatever the changes we want to bring about in a particular plant cell, we can do only in case if the protoplast is available. If protoplast is not here, then automatically whatever the changes we want to bring in, So it's going to be highly difficult to make those particular changes within the plant variety. So first and foremost, we have people actually started doing this particular change of our genetic manipulation is through protoplast fusion by hybridization method. So how this protoplast fusion was being achieved is, people used to develop the protoplast first in the form of suspension culture, like from the callus or from an intact tissues. So for example, if we take intact tissues like leaves, the explant material, so it can be it will be subjected to mechanical disruptions, and by treatment with the cellulitic and as well as pectinolytic enzymes like pectinases, cellulosases and all, which essentially they are going to break the plant cell wall material, that is cellulose and pectins. So because of which then later those particular by the process of centrifugation, all the degradative materials are going to be separated from the cells. So these particular cells are going to be washed continuously with the osmolytes, and then they are going to be means that will give us the protoplast suspension. So once the protoplast suspension is collected through the process of centrifugation and washing in a medium without the enzyme and the only the osmolytes, so then these particular protoplast materials or cells are going to be floated on the sucrose solution, and then when they are plated with suitable medium, nutritional medium, they will synthesize the new cell wall within 5 to 10 days and then initiate the cell division. So that's how actually people used to develop the protoplast. Now how exactly to introduce a gene into the plant? See, in case of animal systems, in case of prokaryotic systems, we know there are a number of vectors which people actually developed for the process of gene transfer. But in case of plants, so there was a hitch in the transfer of genetic material into the plant at the vector systems. Why? Because in case of plants, first they have a cell wall. Second, even if we are going for transfer of genes into a protoplast, so eukaryotic vector systems are not going to hold good for the prokaryotic vector systems will not going to hold good for the transfer of gene into the plant cell. So whatever the vectors which is going to be developed, it should have some of the components, so which is going to able to maintain this particular genes and multiply them within the plant cell. But we have a problem in case of plant cells, that is, whenever we want to go for stable transformation, transients can be obtained by keeping those particular recombinant DNA vector constructs as an episomal structures or extra chromosomal structures within the plant. But if we are looking for a stable transformation, or if we are looking for developing and completely a new plant variety, so an individual plantlet, so then we have an issue, that is, whatever the gene which is going to be transferred, it should get integrated into the genetic material of the plant. Now the problem is, so how to introduce this particular transgene or the target gene into the genetic material of the plant. So for that, people actually studied number of prokaryotic or the plant infecting organisms and cells, and when they have studied various of them, they found so one such kind of bacteria which was infecting the plant and carries in plasmid, so which has some beneficial which actually help the people develop the varieties of vectors for the transfer of gene into the plant system. So once such kind of organism which actually people actually extensively used or harvested for the process of this gene transfer in plants is Agrobacterium tumefaciens. So this Agrobacterium tumefaciens is one such kind of gram positive bacteria found to be present within the soil and infect a plant. So if there is any kind of wound happens at the plant, so then this particular bacteria enters through the wound into the plant cell, plant body, and induces the uncontrolled multiplication of the plant cells at that particular region and creates it and develops a gall, means a mass of tissue, and people call this as crown gall, and they call it as crown gall disease. So when people actually took this particular bacteria and studied it extensively, like why it causes this tumorigenesis or development of the means uncontrolled division of cells at that particular region, how it is doing and all. So then for surprise, people identified that this particular bacteria carries a plasmid called TI plasmid. So tumor inducing plasmid, TI plasmid. So when they studied this particular TI plasmid, then they found this particular plasmid is responsible for actually induction of the tumor, and during which it actually transfers or inserts a part of its own DNA into the plant cell, and by doing so it is going to cause the plant cell to continuously multiply and develops the tumor. So what we will do is we will study about this particular TI plasmids and the derived vector systems of TI plasmids in the next video. Thank you.